Alright, so for my scientific experiment, me and my partners tested the optimal pH value on trypsin activity. To start our introduction, the trypsin enzyme functions in the breakdown of dietary proteins. Its inactive form, trypsinogen, is activated by pancreatic secretory trypsin inhibitors. Trypsin digests proteins by catalyzing the hydrolysis of peptide bonds as well as degrading other proteins. Trypsin can also break down itself, and interruption of this is linked to pancreatitis. The enzyme is vital for health of organisms, since proteins need to be broken down for various functions such as repair and transport. Trypsin is produced by the pancreas in animals and the ribosomes in bacteria. It is found in the small intestine of animals and humans, where its temperature is 37 degrees and its pH is 7.5. We hypothesized that the trypsin activity would initially increase as pH increases, but then eventually denature at a certain optimal value and then decrease. Looking at our methods, our reaction buffers consisted of 0.15 milliliters or 15 m of NaCl for every trial, and then we used different types of pHs, as you can see, 6, 7, 8, 9, 10, 11, which consisted of 20 millimeters of tris hydrochloric acid and 20 millimeters of borate, all at different pHs. We used 15% azocasein, 10% of TCA, temperature baths at 37 degrees, a spectrometer to measure absorbance, a pipette, cuvettes, and centrifuges. In our methods, for each experiment treatment, we used 450 microliters of a reaction buffer, 500 microliters of azocasein, 50 microliters were added to a clean test tube. For the control group, we did not add azocasein, we only used water. Then we added 500 microliters of azocasein and 400 microliters of the reaction buffer. The test tubes were then incubated for 20 minutes at 37 degrees Celsius. And after that, we added 500 microliters of 10% TCA. The purpose of TCA is to stop the enzyme and precipitate azocasein that has not been digested. The test tubes were then put into a centrifuge and at maximum speed for 2 minutes. The purpose of centrifuging after the TCA is to remove the macromolecules of undigested azocasein and the enzyme. Supernatant contains free amino acids and short peptide bonds. The purpose of the substrate is to enable the chemical reaction to form a final product that will be released. Sulfolamide inhibits enzyme activity, which is not correlated to the free amino acids and short peptide bonds. The more free amino acids and peptide chains, the more intense the orange color, which can be measured by the spectrometer at 440 nanometers. We transferred the supernatant to a clean cuvette, blanked the spectrometer at 440 nanometers using a cuvette with water, and read the absorbance of the supernatant at 400 nanometers. When conducting our our experimental variable was the pH of the buffer. The pHs tested were 6, 7, 8, 9, 10, and 11. And the new experiment, we added 9 and 11, where we originally did not have those values, to our experiment. In our control group, we also used 50 microliters or 500 microliters of water for every pH value that we used to compare data with the ones with trypsin and the ones with water. Having a temperature of 37 degrees and 500 microliters of azocasein was our control variables. The 37 degrees regulated our trypsin activity. The temperature also showed enzyme activity at each pH. We used a set of negative controls and completed 3 trials in order to obtain more accurate data. For results, trypsin activity initially increased from 6 to 9, showing it started to denature at 10 and then started to decrease. As you see in this graph, it increases from 6, 7, 8, 9, and then decreases 10 to 11. with optimal ranges of around 11 to 10. Statistical analysis was conducted by a tailed t-test. The overlap of standard deviation bars at pHs 8 and 9 indicate that the activity between these two values was not significantly different. Through the other values, you can see from 6 to 7 and 10 through 11, the data is significantly different with p-values of greater than 0.05. Trypsin activity from pHs 8 to 9 varied by 6%, which is not a lot. So that's why the optimal value could possibly be 8 to 9, depending on more testing. While activity from pHs 9 to 10 varied by 25%, which indicated the enzyme's optimum range of 8 to 9, and a decrease at activity of 10. So for discussion, it was originally hypothesized that levels of pH would increase and the trypsin activity will decrease. After our first experiment, we changed our hypothesis in order to reflect that it would increase and then eventually start to denature and decrease at a pH of 10. Our original hypothesis was not fully supported, as our data showed that it would increase and then would decrease, and we only thought that it would increase, or we thought that the trypsin activity would decrease originally. Our next hypothesis was fully supported, as our results showed that it would increase and then denature after a pH of 10. Our experiment was fully adequate to test our hypothesis. The data based on p-values recorded showed that our optimal value of 9 is significantly different than pHs 6, 7, 10, and 11, but it is not significantly different from the pH of 8. In future experiments, trypsin activity should be tested at more specific pHs relative through pHs 8 through 10 in order to find exactly where the optimal activity of trypsin and denaturing begins. When comparing our data with another group who tested the optimal value of pH, their results were proportional to ours in showing an increase of trypsin activity from pHs 5 to 9 and decreasing at a pH of after 9. Their results show an optimal value of 9 with an absorbance of 0.479, which is very close to our 0.48 absorbance. This pH value concludes that bacteria will grow most efficiently and at fastest at pH of 9. Both groups who tested the effect of pH on trypsin activity also used a control variable of using 37 degrees Celsius as temperature. Other ways to test trypsin activity is the amount of substance you use, solid concentration of a substance, or changing temperatures at which substances are incubated at. The amount of substance used showed substantially lower trypsin activity than other factors, while temperature at 52 degrees reached up to an absorbance of 709, along with a salt concentration of 0.075, NaCl showed a maximum absorbance of 0.680. The certainty of whether the concentration or temperature produces the maximum rate of trypsin activity is unknown due to limiting testing, small difference in values, and the source of possible human error. In lab, there are many factors that could possibly contaminate your data. When pipetting, you must be very cautious and precise, and there's always room to get better at pipetting. A lot of times there are air bubbles that affect this transferring liquids from one cuvette to another. Other sources of variation could relate to getting fingerprints on the cuvette that you use to read absorbance or miscalculations that you could make yourself. The extent to which these things affected our data isn't concerning due to our data being very consistent throughout our experiments and along with the other group that tested pH on the effect of pH on trypsin activity. More replications in the future would help solidify our results and help determine how much these variations affected our results. And here's our works cited.